Today our topic is mechanism of DNA replication and it will be included in the two steps. The first one is unwinding or unzipping and the second one is formation of the new strands. So unwinding means what? Normally in DNA structure we will be having a double stranded and it will be having adenine, thymine and then guanine, cytosine will be bonded with the hydrogen bonds. So here separation of the two strands of DNA that is called unwinding. So these two strands will be getting separated. That one is called unwinding or unzipping. So this process how it will be taking off? This will be due to the breaking of H bonds means hydrogen bonds. So the process of unwinding where it will be starts indefinite in one region and that region we will call as initiation point or origin of replication. So unwinding means what? Breaking of hydrogen bonds. So because of the breaking of hydrogen bonds, we will get origin of replication or initiation point. So this is called unwinding or unzipping. And then see here, in prokaryotes, how many origins will be there? In prokaryotes, only one origin will be there. That's it. In eukaryotes, many origins will be there. That too, all origins will be working at the same time. Means simultaneously, in many places, replication is going on. And for this unwinding and all, means which enzyme is useful for the breaking down of the hydrogen bonds, that is helicase enzyme. So helicase enzyme only useful for the breaking down of hydrogen bonds. Because of this only double stranded DNA will be turned into single stranded DNA and that one we will call replication fork. And with this, along with the helicase there will be a cofactor that is called Mg plus 2. And another one is SSB. SSB means what? Single stranded DNA binding protein. Single stranded DNA binding protein, what it will do means first a helicase enzyme will be unwinding the two DNA strands by breaking down the hydrogen bonds. So afterwards, during the replication, this SSB will be useful for the reformation of H bonds. So, if reformation is happening over there means then again it will be double stranded. So, that reformation will be stopped by the or prevented by the single stranded DNA binding proteins. So, helicase enzyme means what it will do? It will be breaking down of the hydrogen bonds and that separation is maintained by the single stranded DNA binding protein. And remaining few enzymes we will know. First one is topoisomerase. This enzyme is useful for the releasing of tension which is arising from the supercoiling. So DNA will be double stranded supercoiled helix. So that helix will be maintained actually. So from prevention means there will be a lot of tension because of the supercoiling things. So here it is unwinding means supercoiling is stopped. So that supercoiling is released by the topoisomerase enzyme. And this enzyme is present in the eukaryotes. Then what about the prokaryotes means DNA gyrase. So DNA gyrase is useful for the same work in prokaryotes. What is the duration? Minutes. Within a minute, prokaryotic replication will be completed. Within hours, eukaryotic replication will be completed. And the second step is formation of the new chain. So how the formation of the new chain will be there means in the direction of 5' to 3' direction. And which enzyme is useful means DNA dependent DNA polymerase enzyme. So for formation of the new chain or new strand, which enzyme is useful means DNA dependent DNA polymerase. So how many types of DNA materials are there? Polymerase 1, polymerase 2, polymerase 3. Three types are there. And it is discovered by the Korenberg. And what is the use of these three? So DNA polymerase 1 is not at all active, but polymerase 2 and 3 will be having, like, 2 will be having the least activeness in the replication, 3 is having the high more activeness in the replication. So 5 prime to 3 prime polymerization means adding of nucleotides. So this one will be happening in polymerase 1 in a very short chain, that too gap filling work. And in polymerase 2, here also this polymerization will be there, but it is a short gap filling. Same, polymerase 1, polymerase 2, just a gap filling work only it will do. But the DNA polymerase 3, it is having a long gap filling, means most of the work is done by the polymerase 3. And we have to know which one is useful for the removing of the primer after completion of the replication. So that is DNA polymerase 1. So after completion of the replication work, who is doing the stopping work? Means replication is stopped by the polymerase 1. And what is the drawbacks of polymerase 1, 2, 3? Means 3 will be there, but they can't start the replication. They can elongate it. So in origin place, means in the starting place, who will do this work? Means RNA primer. So RNA primer is an enzyme. It is a short chain of RNA that will start the replication. So this started time. After that only replication will be continued by the polymerase 3 in a long chain, polymerase 2 in a short chain, and polymerase 1 in very short gap filling work. And what about the DNA ligase? We will be having DNA newly developed strands in two. One is leading strand, another one is lagging strand. So because of that, the lagging strand is having small pieces of DNA. So that pieces will be joined here and to form a one DNA, that is called DNA ligase enzyme. And next, mechanism. So how the replication will start? Just now we already known about it. RNA primase only is the one enzyme which is useful for the starting of the replication. And how it will be takes place in 5 prime to 3 prime direction only it will takes place. And after formation of the complete new chain, and which will be act as a stopping, means DNA polymerase 1, already we read it. Then what about the formation of the new chain? How it is developed? Because of the nucleotides. Where these nucleotides are present? They are present in the nucleoplasm. Where this process and all will occur? In eukaryotes. Why specifically eukaryotes we are discussing? Means prokaryotes, complete nucleoplasm, cytoplasm, everything is same because it is not differentiated, because it is prokaryote. There is no proper nucleus over there. And in nucleoplasm, nucleotides is there, and that too how it is present means in the form of triphosphates. Example, DATP, DGTP, DCTP, DTTP. So what is this DDDD? This is deoxy and adenine triphosphate, guanine triphosphate, cysteine, and then thymidine triphosphate. And during replication, two phosphate groups will be there, and because of the separation of the two phosphate groups, energy will be released. So when energy is released, that will be consumed during the replication. So when the two DNA strands are getting separated, that time energy will be released. So that energy will be consumed by the DNA replication time. In addition to the DNA dependent DNA polymerase, few enzymes also will be involved over here for the completion of the polymerization of nucleotides. And for the long DNA molecules, since two strands will be there, no? So DNA can't be separated in entire complete length. So there will be a small openings will be there. So that small openings in the DNA we will call replication fork. So in our body, entire DNA can't be opened at the same time and can't be doing the replication at the same time. So in few places, small gaps will be there, and that place only replication will be occur. So how it is occurring means because of the formation of replication fork, unwinding of double stranded DNA by breaking down of hydrogen bonds. Which enzyme is useful for that means helicase enzyme. And that supercoiling will be completely maintained, the tension and all will be released and then maintained. That opening who means that is tropoisomerase. Who is joining the DNA strands means ligase enzyme in Okazaki fragments. Now we will see replication. Like everything is completed. Now we will be having the two strands. One is the leading strand, another one is the lagging strand. Leading strand means what? It's a continuous strand. So this will be developed from 3 to 5 direction. And in the same direction, replication fork also will be developed. Means old strand will be in 3 to 5 direction. In the same direction, new strand also will be here. But in the lagging strand, this is discontinuous strand, and we will call this one, it's a lagging because there is a small fragments of DNA pieces will be here, and newly stranded means newly synthesized one will be in the opposite direction. So these small pieces we will call Okazaki fragments, and those fragments will be joined together by the DNA ligase enzyme. So we already learned that replication will be in semi-conservative. So semi-conservative means what? One strand will be the parent strand and another strand will be the daughter strand. So in every DNA fragment, in every DNA strand, one will be the parent, one will be the daughter. So that's what here half of the material will be conserved. So only this is called semi-conservative replication. Hope you'll understand. If you have any doubts, just comment it.